right, in this screencast, we're going to be looking at the phase of mitosis. But before I get into those different phases of mitosis, I just want to talk to you a little bit about an overview of what's going on during mitosis. First of all, mitosis is the division of the nucleus, but usually accompanied alongside that is cytokinesis, which is the division of the cytoplasm. And what's going on is if this only has one chromosome in it, but we know that human cells have 46 chromosomes. So imagine that there's 46 of these chromosomes, right? And these 46 chromosomes, before mitosis begins, will have replicated during the S phase. When they replicate, so instead of being just a single, chromosome, there's still a single chromosome, but they're actually, there's two copies. And they're attached to each other at a place called the centromere. So this is one chromosome, and this is also one chromosome. And we consider it a single chromosome because it's still attached to the centromere. But this chromosome is different because it's made up of two sister chromatids. These sister chromatids are exact copies of each other. So this chromatid is the exact same copy as, the same nucleotide sequence as this sister chromatid here. And they're held together at a centromere. So when a cell goes into mitosis, it's already in this phase, right? So it's already been copied and it's starting to condense. When mitosis, when it finally starts to split, what's going to end up happening in mitosis is these sister chromatids are going to be pulled apart into two different cells. So at the end of mitosis, we're going to get two new cells, and this one will get one sister chromatid, and this one will get another sister chromatid. Once the sister chromatids have been pulled apart from each other, they're actually considered full chromosomes at that point. You don't need to call them chromatids anymore. So we start with 46 chromosomes, and those get duplicated during the S phase of interphase. And then we end up with 46 chromosomes, but all of those chromosomes are duplicated. Okay, and they're made up, each chromosome is made up of two sister chromatids held together at a centromere. By the end of mitosis, all of those 46 chromosomes are going to split their sister chromatids apart, and we'll get two cells. Each of those cells will have 46 chromosomes, but those chromosomes will no longer be duplicated chromosomes. They will be single chromosomes without the copied sister chromatids. Okay, so we start with one cell of 46, and we're going to end up with two cells of 46. But somewhere in between, there's some copied, duplicated chromosomes that are held together at the centromere. All right, so let's go into the phases. You need to be able to look at these phases and be able to kind of annotate a diagram, recognize the diagram, and talk about what's going on. So you remember interphase is not actually one of the phases of mitosis. This is part of the cell cycle. So we already talked about that. But we know at the end of interphase, at the end of G2, the chromatin, so all those chromosomes are starting to condense. And the chromosomes have already replicated. They replicated during the S phase. And now they're starting to condense. As we move into prophase, so the first actual phase of mitosis, you can see that the chromosomes are starting to fully condense. This is the beginning of prophase, and this is kind of the end of prophase. So you can see the chromosomes continue to condense, and then they're finally condensed. This is called supercoiling. Okay. You can see the nuclear envelope is breaking down. Okay, so here's the nuclear envelope in the beginning, and by the end, it's fully broken down. The centrosomes. So these are called centrosomes. In animal cells, the centrosomes have these little things called centrioles, but plant cells don't have centrioles. So I tend to call them both. I just call it the centrosome area. So this is a centrosome. This is a centrosome. And the centrosomes are going to start to migrate to either end of the cell. So you'll see by the end of prophase, one centrosome is here and the other centrosome is there. The centrosomes are responsible for making the spindle, the mitotic spindle. So you can see the spindle fibers are starting to form here. And then by the end, the spindle fibers will have attached to each of the chromosomes. And you can see on the chromosomes, they are attached at the centromere. So that's the middle of the chromosome here where those two sister chromatids are attached. And you can see, just looking in this one, a spindle fiber from this side of the cell is attached to this side of the centromere, and a spindle fiber from this side is attached to this part of the centromere. Okay, so once they're all attached, As you saw in the animation before all this started, things jig around a bit, and they end up pushing and pulling. All these spindle fibers push and pull back and forth until all of the chromosomes in metaphase are lined up along the metaphase plate. Okay, so you're going to get one side of the chromosome is going to go this way, and the other side of the chromosome, that other sister chromatid, will go this way. All right, so here we have the 46 chromosomes will all be lined up along here. This is called the metaphase plate. As you enter anaphase, what happens with anaphase is you get the spindle fibers are going to actually, the ones that are attached to the chromosomes at the centromeres, they actually start to shorten, and that's going to pull the chromatids apart. The other spindle fibers, these guys, are going to continue to lengthen, which is actually going to push the poles of the cell to, apart from each other, get them further and further apart. And so you're going to see these start to migrate further up because these ones are shortening and these ones are lengthening, and you're going to pull the chromatids. 46 of the chromatids go this way, 46 of the chromatids go this way. And actually, once they've separated from each other at the centromere, we can now call them chromosomes. They're kind of like, it's like they graduated. They're full-fledged chromosomes now. And then finally, when we get to telophase, move my head, when we get to telophase, the nuclear envelope will start to reform. You can see the nucleolus starts to reform. The mitotic spindle is, breaking down, and the chromosomes will start to uncoil. And in the end, we're going to end up getting two fully formed cells that both have 46 chromosomes in them. All right, so mitosis is the division of the nucleus, but also part of the M phase usually is cytokinesis. So let's take a look at cytokinesis. And cytokinesis in plant cells and animal cells is slightly different. In animal cells, you get what's called a cleavage furrow. And in this, if this is, the two cells, the cytoplasm actually starts to pinch off because the cell membrane is pretty fluid. So it's able to create this contractile ring of microtubules, and it starts to pull together, and then finally it just kind of divides and the cell membrane reforms on either side. It's pretty simple. They call that cleavage of the animal cell. A plant cell, on the other hand, though, because it's got the cell wall, it is a little bit more complex. What happens is that from the endoplasmic reticulum, a lot of these little vesicles that contain pieces of cell wall will start to migrate towards the middle of the cell. We call that the cell plate. So there's these coming from both of the new daughter cells. There's vesicles that contain pieces of cell plate that will start to come towards the middle. And the vesicles, of course, have a cell membrane around it. And then that cell membrane will end up fusing on either side, and the pieces of the cell wall that they were carrying can fuse together to make a new cell wall. So then that cell wall, those pieces of the membrane that were in the vesicles become the membrane on that side and the membrane on that side. And then there's cellulose that forms that cell wall within the middle.